Assalamu alaikum everyone. Today is my first lecture on Human Genome Project. It was published in 2003. We will start from the history of Human Genome Project. So in 1977, Fred Sanger sequenced the genome of bacteriophage. It was 5,375 base pairs. It was the first genome ever sequenced. Then in 1985, Robert Sinsheimer proposed the idea of sequencing the entire genome. Many scientists considered the project exciting, but it was impractical at that time. Later in 1980s, the idea faced criticism because it was expensive and technologically difficult. The American Academy of Science recommended a gradual approach. What was that approach? That was sequence simpler organism first, develop better sequencing technologies, and then sequence the human genome. In 1988, US Congress approved funding. NIH published the Office for Genome Research under James Watson. In 1989, Human Genome Organization HUGO was founded to coordinate international genome research. Early in 1990s, Craig Venter developed expressed sequence states ESTs for rapid gene identification. A controversy arose over patenting genes. What does patenting genes mean? Basically, it is ownership of an invention. If a scientist patent his work or discovery invention, so it's mean that when any other scientist wants to use that in his work, he have to ask for permission or to pay for that. So Craig Venter opposed application that to patent his work like ESTs. So this is what patenting genes mean by. Then in 1992, James Watson resigned due to disagreement over gene patenting. Craig Venter left NIH and started private genome research. Due to opposition, Craig Venter got separated from NIH and started his own private genome research. Then in 1994, Harold Warmus, he was the director or the head of NIH. In 1994, he decided not to pursue gene patent applications, like he rejected or opposed this application. Then in 1995, Craig Venter and colleagues sequenced the genome of Haemophilus influenza. It was the first free-living organism to be completely sequenced. This was sequenced through a shotgun sequencing method. In 1996, NIH funded scientists completed the genome sequence of Becker's yeast, Saccharomyces cerevisiae. Then in 1998, Craig Venter founded Celera Genomics. It was his own company. He announced that the human genome, would be sequenced within 3 years using shotgun sequencing method. Then in 2000, Celera claimed to have mapped 90% of the genome. The public Human Genome Project also reported major progress. On June 26, 2000, a joint announcement of the draft human genome was made by Craig Venter and Francis Collins. Then in 2001, Celera's Human Genome paper was published in Science, and in 2003, the Human Genome Project was completed, providing full sequence of human genome. So this was all about the Human Genome Project. I hope that you get benefit from this lecture. And if you want more videos, handwritten notes, then you can write it in the comment. Soon I will upload a lecture on that. And at the end, I want you to like, share and subscribe my YouTube channel. It's my first video and I want to grow a community and where both of us get benefit. So thank you so much for watching this video and stay with me. Thank you.